Severe acute respiratory syndrome, SARS, caused by SARS coronavirus, triggered a global epidemic spanning 32 countries in 2003. Symptoms include fever, liver damage, and respiratory failure. There is currently no cure, but treatments include antibiotics, steroids, and ventilation support. PLpro, a necessary component of SARS viral replication, is a cysteine papain-like protease. PLpro is an active site that consists of a catalytic triad with residues cysteine 112, histidine 273, and aspartate 287. The cysteine thiolate cleaves peptide bonds of substrates. PLpro not only aids in viral replication through its interaction with the polyproteins, but also hinders immune response through the hydrolyzation of immune proteins ubiquitin and ISG15. Recently, GRL0617 has been shown to inhibit PLpro and SARS-CoV, leading to the inhibition of viral replication. It also decreases the spread of infected cells through hydrolysis of ubiquitin and ISG15. In a 2007 study by Chen et al., SARS-CoV was compared to the human coronavirus HCoV-NL63 to investigate its replication mechanism. This study was done because the two CoVs share identical replicase products in similar proteases, PLp2 and PLpro.
Both CoVs utilize the ACE2 receptor to infect the host cell through endocytosis. Once inside this host cell, the CoVs release genomic RNA into the cytoplasm. Replication of viral RNA begins. Translation machinery of the host cell translates the overlapping ORF1a and ORF1b to produce polyproteins pp1a and pp1ab. PLpro processes these polyproteins to form the replicase complex, which leads to the transcription of subgenomic mRNAs for 16 viral proteins. These non-structural proteins, NSPs, assemble with the endoplasmic reticulum membranes to form the replicase complex, which helps replicate the virus's RNA and spread infection. Proteins M, S, E, and N are formed from transcription of genomic RNA. These proteins then undergo modification at the Golgi and interact with one another to form a budding virion. The virions are then exported and released to infect other cells. PLpro is also involved in hydrolyzing ubiquitin and ISG15. Besides the active site, PLpro has two other ubiquitin binding subsites, which gives SARS-CoV PLpro additional specificity to ubiquitin. Because PLpro proves to be vital in SARS-CoV replication and protection against immune response, it has been targeted in research. Several inhibitor classes for SARS-CoV PLpro have been identified. Most recent studies focuses on naphthalene inhibitors.
Specifically, in a 2008 study by Ratia et al., the competitive naphthalene inhibitor GRL0617 has been shown to inhibit PLpro function and impede viral replication in viro E6 cells. After testing over 50,000 potential inhibitors, the most potent was structurally altered to optimize the inhibitor and obtain the lowest IC50 value. This value is the inhibitor concentration at which protein activity is halved. The lower its value, the better the inhibitor. This new optimized inhibitor was named GRL0617. GRL0617 binds non-covalently to the S3 and S4 subsites in a cleft near the active site of PLpro. The inhibitor's amide group forms two hydrogen bonds with aspartate 165 and glutamine 270 in PLpro and engages in hydrophobic interactions with nearby residues. The inhibitor alters the conformation of PLpro's active site and oxidizes the cysteine residue in PLpro's catalytic triad. Thus, PLpro loses its ability to cleave SARS-CoV polyprotein and form the NSPs for the replicase complex. Deubiquitinating enzymes can be labeled by HAUB vinyl sulfon, an active site probe. If labeled, the protein can be shown in a gel using a Western blot. PLpro, a DUB, was tested with and without GRL0617. When no inhibitor was present, the PLpro was able to be labeled, and thus it could be visualized on the gel. In the presence of inhibitor, the band is absent, which shows that GRL0617 was bound to PLpro. The inhibitor was tested in viro E6 cells against PLpro in 4 human DUBs. The results showed that GRL0617 prevented PLpro from carrying out its reaction, but did not inhibit human DUBs at all, because they contain phenylalanine 409 and lysine 420 residues that cause steric hindrance. Thus, GRL0617 demonstrates specificity for PLpro. Despite its inhibitory activity in preliminary studies, GRL0617 is not potent enough to be used in treatment. However, the compound provides a basis for future studies on naphthalene inhibitors. Future studies may include clinical trials with SARS-CoV PLpro inhibitors to test their efficacy in biological conditions.